Have you ever taken an antibiotic and just felt better afterward? It's a modern medical miracle we often take for granted. But imagine a world without them, a world where a simple scratch could lead to a deadly infection. That world was a reality just over a century ago. Today we're diving into the incredible story of a happy accident, a messy lab, and a revolutionary discovery that changed medicine forever. The story of penicillin. Our story begins not in a pristine high-tech laboratory, but in the slightly cluttered workspace of a Scottish scientist named Alexander Fleming. It's the summer of 1928, and Fleming, like many Londoners, is getting ready for a holiday. Before leaving, he had been studying Staphylococcus, a common type of bacteria responsible for everything from sore throats to life-threatening abscesses. He had a stack of petri dishes filled with these bacteria, and in his haste to leave, he didn't quite clean up properly. He left the dishes stacked on a bench in a corner of his lab. When Fleming returned 2 weeks later, he started the tedious task of cleaning up. As he sorted through the dishes, one caught his eye. It looked different from the others. A mold, a fuzzy blue-green patch, was growing on one of the dishes. Now, for most people, mold on old food is just a sign to throw it in the bin. For a bacteriologist, it usually meant a contaminated, failed experiment. Fleming was about to toss it, but something made him pause and look closer. Around the patch of mold, there was a clear, halo-like zone where the Staphylococcus bacteria had been completely wiped out. It was as if the mold had erected an invisible barrier, a kill zone that the bacteria couldn't cross. The bacteria further away from the mold were growing just fine, but right next to it, they were destroyed. Fleming was intrigued. This wasn't just contamination. This was something more, he famously said. That's funny. It was a classic understatement for a discovery that would save millions of lives. Fleming realized he had stumbled upon something incredibly important. He took a sample of the mold and began to cultivate it. He identified it as belonging to the Penicillium genus, so he named the substance it produced penicillin. He then conducted a series of experiments. He found that this mold juice, as he sometimes called it, was remarkably effective at killing a wide range of harmful bacteria, including those that cause pneumonia, meningitis, and diphtheria. What was even more astonishing was its effect on living cells. He tested the crude penicillin filtrate on white blood cells from a human. Unlike harsh chemical antiseptics of the time, which killed bacteria but also destroyed the body's own defensive cells, penicillin was non-toxic to human cells. It was a selective weapon, a magic bullet that targeted the invaders while leaving the defenders unharmed. This was the holy grail of infection treatment. Fleming published his findings in 1929. You'd think the world would have immediately hailed him as a hero, right? Well, not exactly. The scientific community was largely unimpressed. Fleming was a bacteriologist, not a chemist, and he struggled to isolate and purify the active ingredient, penicillin. The crude broth he worked with was unstable and lost its potency quickly. It was also difficult to produce in large quantities. Couldn't get enough of it to even treat a single infected mouse properly, let alone a human patient. For about a decade, Fleming's groundbreaking discovery sat on a shelf, a fascinating academic curiosity but not a practical medical treatment. Fleming himself moved on to other research, though he always believed in penicillin's potential. Often sharing his mold cultures with other scientists who requested them, hoping someone could unlock its full power. The story could have ended there, but then World War II began. The war created an urgent, desperate need for a way to treat infected wounds, which were killing more soldiers than bullets were. The call for a new weapon against infection was louder than ever. This is where two new heroes enter our story: Howard Florey and Ernst Boris Chain, a team of scientists at Oxford University. In the late 1930s, they came across Fleming's old paper. Intrigued by the potential of a substance that could kill bacteria without harming human cells, they decided to take on the challenge that Fleming couldn't overcome: purifying penicillin. Their work was painstaking and underfunded. They started with a small grant and used an odd collection of lab equipment, including bedpans and milk churns, to try and grow the Penicillium mold in larger quantities. Chain, a brilliant biochemist, finally succeeded in isolating a concentrated, stable brown powder of penicillin. It was a tiny amount, but it was enough for the next crucial step: animal trials. In 1940, they conducted a landmark experiment. They infected 8 mice with a lethal dose of Streptococcus bacteria. 4 of the mice were left untreated. The other 4 were given injections of their purified penicillin. The results were dramatic and unequivocal. The next morning, the 4 untreated mice were dead. The 4 mice that had received penicillin were all alive and well. Florey and Chain knew they were onto something monumental. They'd proven that penicillin worked inside a living body. Now, they needed to see if it worked in humans. Their first patient was a 43-year-old policeman named Albert Alexander. In late 1940, a simple scratch from a rose thorn on his face had become severely infected. The infection had spread, causing abscesses all over his head, and he was close to death. Doctors had tried everything, but nothing worked. In February 1941, as a last resort, he was given penicillin. The effect was almost miraculous. Within 24 hours, his fever dropped, and the infection began to recede. He was getting better, but there was a problem. They were running out of penicillin. The team at Oxford was working around the clock, even collecting the penicillin excreted in their patient's urine to repurify and re-inject it. But it wasn't enough. After 5 days of treatment, their supply ran out. The infection returned with a vengeance, and sadly, Albert Alexander passed away. While a tragedy, this case proved 2 things. Penicillin worked in humans, and they needed to find a way to make a lot more of it. With Britain under constant threat of bombing, mass production was impossible there. So, in the summer of 1941, Florey and a colleague flew to the United States with a secret mission to convince American pharmaceutical companies and the US government to back the mass production of penicillin. They carried their precious mold sample with them, reportedly smearing it into the lining of their coats to make sure it survived the journey. In America, they found the industrial might they needed. Scientists at a government lab in Peoria, Illinois, made crucial breakthroughs. They discovered a new, more productive strain of Penicillium on a moldy cantaloupe found at a local market. This strain, dubbed the Peoria strain, produced over 200 times more penicillin than Fleming's original. They also developed a new fermentation method, using deep vats and corn steep liquor, a byproduct of corn processing, to grow the mold on an industrial scale. With the full backing of the US government, pharmaceutical companies like Pfizer and Merck retooled their factories. By D-Day in June 1944, enough penicillin was being produced to treat all the Allied wounded soldiers. It was called the Wonder Drug, and it dramatically reduced deaths and amputations from infected wounds. Its impact was so profound that some historians argue penicillin saved more lives during the war than were lost in combat. After the war, penicillin became available to the public, and the age of antibiotics began. For their incredible work, Alexander Fleming, Howard Florey, and Ernst Chain were jointly awarded the Nobel Prize in Physiology or Medicine in 1945. Fleming, ever the humble scientist, always gave credit to Florey and Chain for turning his chance observation into a life-saving medicine. He referred to his role as simply a matter of nature making penicillin, and he just happened to find it. The discovery of penicillin is a perfect example of how science often works, a combination of chance, curiosity, perseverance, and collaboration. It started with a messy lab and a curious mind, was ignored for a decade, and then revived by dedicated researchers who refused to give up. Their collective effort gave humanity one of its most powerful tools against disease and ushered in a new era of medicine. It's a reminder that sometimes the biggest breakthroughs can come from the most unexpected places. Thanks for watching. If you found this story as fascinating as I do, please hit that like button. Subscribe for more deep dives into the history of science and medicine. See you in the next video.